Welcome to this video. Today, I'll create 10 competitive exam-style multiple-choice questions about amino acids, peptides, and proteins, complete with detailed explanations. So, let's dive right in. Alright, let's start with our first question. Which of the following amino acids contains a sulfur atom in its side chain? A. Lysine. B. Methionine. C. Threonine. D. Aspartate. The answer is B. Methionine. Methionine contains a thioether or SCH3 group in its side chain. Cysteine is another sulfur-containing amino acid, but it has a thiol or SH group. Moving on to our next question. At physiological pH, which is 7.4, which amino acid would have a net negative charge? A. Arginine. B. Lysine. C. Glutamic acid. D. Histidine. The answer is C. Glutamic acid. Glutamic acid has a pKa of approximately 4.1 for its side chain carboxyl group. At pH 7.4, this group will be deprotonated, giving the amino acid a net negative charge. Now, let's tackle the third question. The peptide bond is A. Highly flexible and freely rotating. B. Rigid and planar. C. Always in CIS configuration. D. Easily hydrolyzed under physiological conditions. The answer is B. Rigid and planar. Due to resonance stabilization, the peptide bond has partial double bond character, making it rigid and planar with restricted rotation. Which level of protein structure refers to the specific three-dimensional arrangement of alpha helices and beta sheets? Is it A. Primary structure. B. Secondary structure. C. Tertiary structure. D. Quaternary structure. The answer is C. Tertiary structure. Tertiary structure describes the overall three-dimensional folding of a polypeptide chain, including the spatial arrangement of secondary structure elements. A tripeptide consists of A. 3 amino acids linked by 2 peptide bonds. B. 2 amino acids linked by 3 peptide bonds. C. 3 peptide bonds only. D. 3 amino acids without peptide bonds. The correct answer is A. 3 amino acids linked by 2 peptide bonds. A tripeptide is formed when 3 amino acids are joined by 2 peptide bonds in a linear sequence. The R group of which amino acid is most likely to be found in the interior of a globular protein? Is it A. Lysine. B. Leucine. C. Glutamic acid. D. Asparagine. The answer is B. Leucine. Leucine has a hydrophobic side chain and tends to be found in the protein interior away from the aqueous environment. 7. Which technique separates proteins primarily based on their molecular weight? Is it A. Isoelectric focusing. B. SDS page. C. Ion exchange chromatography. Or D. Affinity chromatography. Answer B. SDS page. Explanation: SDS page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis, denatures proteins and separates them based on molecular weight. 8. The isoelectric point, or pI, of an amino acid is A. The pH at which the amino acid has no net charge. B. Always equal to pH 7. C. The pH at which the amino acid is most soluble. Or D. The pH at which the amino acid is fully protonated. Answer: A. The pH at which the amino acid has no net charge. Explanation: at the isoelectric point, the amino acid exists as a zwitterion with no net electrical charge. 9. Which of these bonds is not typically involved in stabilizing protein tertiary structure? Is it A. Hydrogen bonds. B. Peptide bonds. C. Disulfide bridges. Or D. Hydrophobic interactions. Answer: B. Peptide bonds. Explanation: peptide bonds form the primary structure. The tertiary structure is stabilized by hydrogen bonds, disulfide bridges, hydrophobic interactions, and ionic bonds. 10. Denaturation of a protein. A. Breaks peptide bonds. B. Changes primary structure. C. Disrupts higher order structure. Or D. Converts amino acids to different forms. Answer: C. Disrupts higher order structure. Explanation: denaturation disrupts secondary, tertiary, and quaternary structures while leaving the primary structure, which are the peptide bonds, intact.